So I'm Jordan. I'm one of the GMods exec members. I'm going to give the newsreel today. Also, Happy New Year. First GMods meeting of the new year. It's exciting. Okay, so first community news. We have 2 new podcast episodes. First, we interviewed Dr. Carmela Haynes, who's a professor at Arizona State University. She does engineering of chromatin, which is super cool. She's like one of the only people that's really working on that. And then our most recent episode, we've split them up into interviews and new, like Wait, Wait, Don't Tell Me style quiz formats where we talk about the news. And we had Isaac and Adam Silverman and me do a little news quiz. Adam and Patrick Donahue are also the other 2 exec members. They'll be here in a second. So we have 2 new podcasts. We'll send out links to that again. Isaac had another announcement about the Free Genes project. Do you want to talk about that instead of me, since I forgot?
No, it would really be tooting my own horn, so it would be really embarrassing.
Okay. So there's a project out of Stanford with Drew Endy called now it's called the Free Genes Project, which Isaac has become very involved in. They have for the first time figured out how to ship their plasmids that they're sending out to people. So the Free Genes Project, to backing up a little bit, is they're trying to come up with a repository of 10,000 free genes for like any researchers, startups, whoever to use. And basically you can just suggest one, and now they're going to ship it to you, and they've come up with this cool way to ship them using Bacillus subtilis spores. Is that right, Isaac?
Yep, Bacillus subtilis.
And they've tested that for the first time, and it apparently works really well, and Isaac was one of the people who helped test that in his lab.
And Callus too.
And Callus, yeah. So that's super cool. More community news. So getting into other news, I always like to include in the newsreel a popular science article that you can share with your Muggle friends who are not scientists. So this is a cool article that came out in the New York Times. This is about David Baker's lab and designing proteins from scratch. Oh, thank you. So it talks all about David Baker's lab and sort of evolving proteins that aren't found in nature. It talks a little bit about, and I'll talk about, there's more David Baker news at the end of the newsreel. So it talks about some of the proteins they're making, like proteins that are like flu vaccines, lots of cool stuff there. So definitely show this article to your friends who are not in science. Industry news. Ginkgo Bioworks has become the first synthetic biology unicorn, which is a startup that's valued over $1 billion. This is actually the first, when you Google image search unicorn, this is the first picture that comes up. So yeah, Ginkgo has become the first unicorn in the synthetic biology industry, and that's so synthetic biology on the up and up. That's getting more venture capital money and everything. It's pretty cool. There's this interesting new project coming out of Y Combinator, which is sort of a startup incubator thing that funds startups. That they are coming up with this thing called YC Bio, which funds startups that are still in the lab phase, that are very, very small, that they're focusing on companies that are have a focus on human longevity and aging. So that's something that Sam Altman is working on. This is sort of industry biohacking news. I think this was sometime in December. Also, since we didn't have a December meeting, this includes some December and January news. The FDA announced a statement that ostensibly came off the tail of a couple of people whose names shall not be mentioned, who have been testing testing gene therapies on themselves. So I will not mention his name. So this guy, Josiah Zahner, I'll mention his name. injected himself with a gene to knock out myostatin, which is a protein that inhibits muscle growth. So you knock it out, and essentially your muscles will get bigger. That's probably not actually going to happen. So, so the FDA and several other people have been doing this and testing gene therapies on themselves. The FDA has issued a statement, actually not that that is illegal, but that the sale of gene therapy kits for self-administration is illegal. So something else that Josiah Zahner's company, the Odin, I think is selling kits that include the myostatin gene, but it says don't actually inject this with yourself, but essentially if you wanted to, you could. Yeah.
It's for research purposes.
For research purposes. So the FDA had sort of a harsh statement that was like, untested gene therapy, this is illegal, don't do it. And there were a lot of articles that came out about that, and some of the biohacking community who's pro self experimentation was like, we're going to do this anyway. So that is an interesting development on the biohacking front. So other industry news. Bolt Threads has come out with another new product, which is a hat. So they have a new beanie line that they only made 100 of them, and they sold out like instantaneously. But it's made out of synthetic spider silk, so bioengineered spider silk. That it's actually a blend of spider silk and wool, so it's soft and durable. So they also got a ton of venture capital money recently. So Bolt Threads is doing really well. So Biomaterials also getting a good start in end of last year and the new year. Now into research news. This is actually a paper from, this is a news brief for a paper that's in October, but I thought it was really cool, so I included it anyway. I also thought this was a great lead. Mesiej Veselko has made wild sex deadly for genetically modified organisms. Yeah, so this is an interesting project that they've basically taken yeast and made two different sort of mating types synthetically. So they use CRISPR to basically mutate an actin gene. So and in one of the strains, and in the other mating strain, they have sort of a CRISPR antidote toward it. So in the one strain, actin sort of like mutates and grows uncontrollably. So as you can see in this right panel, if they don't mate with the mating strain that has the antidote, they will just sort of grow out of control and explode. But if they do have the correct mating strain, then they will grow normally. So this means that this synthetic sort of yeast system can't mate with wild yeast strains. So this is a good development for sort of preventing like environmental spillover and sort of synthetic biology strains escaping into the environment. So this is another cool paper.
Seems great for like studying gene drives.
Exactly. Yeah. Good system for gene drives. This is also really cool. So nanopore sequencing has done RNA for the first time without needing reverse transcriptase and cDNA. So if you don't know about nanopore sequencing, basically it uses these protein channels to feed sort of long DNA transcripts through this little pore, and it measures the changes in current based on the different nucleotides. So the change in current is slightly different for each nucleotide, and you can measure that and then get a sequence readout. So they've been able to do this on RNA for the first time. So this should make RNA-seq a lot easier and faster. MAGE has been used in yeast for the first time. This came out at end of November. We didn't have a newsreel in November either. So MAGE is a sort of recombinant, or not recombinant, a sort of gene editing kind of thing used to basically it knocks out endogenous DNA repair mechanisms in organisms and allows them or keeps them from. Someone just explained to me. I did not know what MAGE was before this, so bear with me. Sorry, I also put this together kind of late.
It fixes the mismatch repairs.
Fixes the mismatch repairs, so you can put in specific, or you can put in like random mutations into the DNA, and it won't fix it. So you can basically come up with mutations throughout the genome that you would want. So it's basically been used a lot in bacteria, but they haven't been able to use it in eukaryotes and yeast, but they've been able to do that for the first time in this paper. Here is the David Baker coming back again. This is from the Baker lab. So David Baker makes computationally designed proteins. So this is really interesting. They have made a sort of protein cage, kind of an artificial, almost like a viral capsid that encapsulates its own RNA. And one of the cool things about this is that they were able to get the RNA loading into the protein cages that was approximately comparable to the efficiency that you would get loading RNA or nucleic acids into viral capsids, which is about 10%, which is kind of low, but apparently that's the benchmark that people actually use. And then this is my last one. I thought this was kind of interesting. People are using, are sort of using biology to manufacture indigo, which is used as a dye for blue jeans. And something, in order to actually dye fabrics with indigo, you need to use like a harsh reducing step. And this group figured out a way to include a sort of a glucose residue onto the sort of indigo, or the precursor to the indigo molecule that they were able to then sort of remove in the dyeing step that created some, allowed the whole process to be a lot less harsh. So this will be a very sort of a more environmentally friendly way to make fabrics, or dye fabrics, which is pretty cool. So that's all I have. We're gonna wait for a second and get pizza, and then Sarah Stainbrook is gonna talk about engineering plant synthetic biology. Okay, I will go see what's up.
Okay, so I'm just going to get started and we'll break for pizza whenever it gets here. So Isaac kind of sent me a whole bunch of links, none of which I actually used, so he has them to send them out to you guys if you're curious. So building plants of the future, I realize, could kind of be taken two ways. I am not looking at building futuristic plants, which is what Isaac can send out, the synthetic blue rose, or a lot of the work by Jim Medford at Colorado State. I'm looking more at building plants to prepare us for the future. So today, I kind of wanted to focus on looming global problems and how plants can help us fix them. So the three I'm going to focus on are pollution, climate change, and malnutrition, and how plants can solve them through phytoremediation, stress tolerance, and biofortification. And so I'm just starting to get into this field of plant engineering, plant synthetic biology, and you think of greenhouses, but I never really thought about little seedlings in a test tube, so I'm going to be showing pictures of what these actually look like in the lab, and still trying to get used to it. Okay, so starting very basically, where do plants come from? So a long, long time ago, one bacteria ate a photosynthetic cyanobacterium that turned into a chloroplast, and now we have plants. Basically. So these diverged from all other forms of life a very long time ago, which means that they are very different from mammalian, bacterial, or even fungal cells, which gives them some interesting and sometimes challenging properties. So here are some of the differences between the organisms we're used to working with and plants. Obviously, plants are photoautotrophic. They create their own energy from sunlight or other light, and so they don't need to be fed glucose or any other energy source. They do need micronutrients from the soil, nitrogen, phosphorus, potassium, those sorts of things. But generally, the energy component comes through sunlight. They have a cellulose cell wall. They're very rigid, so fungal cell walls are somewhat rigid. These are much more rigid. Their development is a lot less constrained. So we think of mammals, you've got a head, arms, legs, they need to go all in the right places. We have these very rigid developmental protocols that come through gene regulation. Plants don't care. Plants just don't care. Whether you have 2 leaves coming out like this or, another branch over here, their development is so much less constrained, which makes it a lot more flexible what you can do to them in terms of mutating these developmental genes and still have them survive. They have very different ploidy, so all the organisms we work with are either haploid or diploid. Plants have an even number of chromosomes. I think the highest is 216 ploid in ferns. But for example, wheat is hexaploid, so anytime you try and genetically engineer wheat, you have to change all 6 copies instead of just 2. Almost all cells are totipotent, so if you take a cell from a plant and you stick it in a petri dish, it can regenerate the entire plant. So the technique here, this is called Callus. Basically, a single genetically modified cell was put in this plant, and it grows this undifferentiated tissue mass, kind of like a tumor you'd think for a mammal or a eukaryotic. You know what I'm trying to say. And then it can differentiate into the shoots, the roots, everything else. And obviously, plants are stationary. They don't move. Now, that's kind of a no-duh thing, but it's actually really important because that means that it's constrained to deal with whatever conditions it has in whatever location it is. It can't get out of the weather. If there's a contaminant spill, it can't get out of that. And so they've developed a lot of these strategies for dealing with environmental conditions that no other organisms really have because they can move. Or sporulate or something like that. So this leads to some differences in engineering. Because light is the food source, these plants aren't terribly permeable to other nutrients. So it's difficult to just apply a like IPTG or something to create this genetic change or change the genetic program. So it's very hard to alter metabolism by outside intervention. Because they have to have these metabolic programs for detoxifying compounds, creating new compounds, stress response, plant metabolism is very complicated, even between different families, but so it's a lot more complex. And they're very situation specific as well. So just growing it in the lab, you won't necessarily find all of the different compounds that it can make or the pathways that are available. Plant transformation is hard because of the cell wall. The typical method is microprojectile, which is a gene gun. You literally cover these gold particles in DNA and shoot it through the cell wall and hope it makes it into the nucleus. Agrobacterium is another way. So these are laboratory tractable bacteria that you can genetically modify, and then you literally dip the seed head into this solution of bacteria, and the DNA gets into the germ cells, which sounds easy, kind of. There's also protoplast electroporation, where you strip the cell wall off and then shock it. The problem with this is that all of these cause random insertions into the genome. In plants, we're a lot further behind in being able to make targeted changes to the genome, with the exception of CRISPR, which is proving very promising, but of course there's all sorts of licensing and intellectual property disputes going on. What's really interesting is that altering ploidy is a valid engineering strategy. So things that you can take 2 diploids and make them into a tetraploid, it can actually improve the stress resistance qualities just by having 4 chromosomes instead of 2. And as I mentioned before, any single cell can produce an entire plant. And so you don't need to specifically transform the germ cell. If you happen to get it onto the leaf and make it into a leaf cell, you can grow an entire plant from that. So these are kind of some very different ways of thinking about how to approach a problem. And I'm going to show one thing about CRISPR, but by and large, to date, everything has been done by this random genomic insertions, which of course you have to worry about regulation. What is the surrounding sequence context? Are you disrupting another gene? Things like that. So starting out, phytoremediation. This is kind of one of the answers to our global pollution problem. It's treating pollutants or waste, contaminated soil or groundwater using green plants that can remove, degrade, or stabilize these toxic substances. So there are some advantages to this. You can just do it in situ. You can plant these plants wherever your contamination is, or you can take your contaminated dirt, for example, move it to another site. Plants have so far been able to break down a large variety of organic and inorganic compounds, which comes again from being evolved to live in one place and having to deal with the conditions there. You can just kind of broadcast these seeds so that you can treat large areas at once, which makes it very cost effective compared to chemically treating to break down a specific chemical or things like that. It's fairly easy to implement. It looks pretty, the public likes it. And because you're using the root systems of these plants to stabilize the soil, you're not going to get leaching or runoff as much as you would with typical treatments. It conserves natural resources and it's environmentally friendly. Unfortunately, it also takes a while. And it's limited to as far deep as your roots can go. So with things like sunflowers, that's maybe a foot. With things like poplar trees, which I'll talk about in a little bit, you're getting closer to 30 or 40 feet. So when you're looking at really deep contamination, this is not going to work. Obviously, you're still subject to toxicity for the plant. So if you really have a very contaminated site, you may not be able to grow plants there anyway. It's slower, it takes several years. Sometimes the products, the toxic substances aren't terribly bioavailable. So they may be toluene, for example. It's not really soluble. And whatever this toxin gets turned into by the plant metabolism, that still hasn't been studied to tell whether that itself is toxic or bioavailable or broken down. And of course, you have to worry about the climate. It doesn't work in the winter. And if you have these plants that suck up the contaminants, then you just have moved your contamination from the ground into these plants. So, but it is one option, and I'm going to show you a couple ways that it has worked so far. So specifically for sucking up toxic compounds, it works typically through these three steps. First, you uptake it and transport it into the cell. And then you transform it from, this is TNT, by the way. Yeah, so you take this TNT and you chemically activate it using an enzyme like cytochrome P450 or carboxylesterases. Then you conjugate it to an endogenous molecule, glucose, something like that, which reduces the toxicity by excluding it from certain biological processes. And you can do that using glycosyltransferases, glutathione S transferases. And then typically, because these plants don't want to put it back in the soil because then they'll just take it up again and can't really get rid of it any other way, they either stick it in the vacuole, which is kind of your waste compartment, or in the cell wall. So you basically just hold on to it, detoxify it, and store it. So one example from very early, I believe this was 1994, this is a pond near Chernobyl. So there were extremely high levels of radioactive compounds, specifically selenium and cesium. These sunflowers and several other types of plants are actually able to suck up these compounds preferentially and concentrate them 2,000 to 8,000 fold what it would be in the water. So they planted these sunflowers on these cute little rafts floating in the radioactive pond. And then they harvested the sunflowers, burnt them, and quarantined the ash because it's that radioactive. So metals aren't terribly bioavailable. And so some of the strategies that are used in plants that don't naturally take up these metals would be to add chelating molecules like siderophores, things that go and scavenge these, collect them, and get re-imported. Metal transporters, biosurfactants can actually go into the soil and increase the bioavailability of metals that are otherwise compounded with other things. And acidifying compounds can change the ionic state of the metals and make them more easy to absorb. And then the metals themselves can be very toxic, and so expressing reducing enzymes within the cells allows the cells to tolerate them better. But yeah, this has been going on for a very long time. In fact, in Fukushima, there are many efforts to distribute sunflowers widely and then tell the people to bring them back. Because if you're collecting radioactive material, you need to dispose of it properly. But that has primarily been the biggest issue, is getting people to bring the sunflowers back. So this one we talked about was just phytoextraction here, where you take it out of the soil and store it. There are other methods. So phytodegradation takes your toxic product, breaks it down. Phytovolatilization takes your compound from the soil and puts it into the air, which is not really ideal, but it happens. And then phytostabilization takes your highly volatile compound and stabilizes it so that it's not going to go into the air. And phytostimulation is where, instead of taking up the compound, you actually secrete something to break it down in situ. So I'll show a few examples of those. But first, rhizome remediation, which is kind of a subset of phytoremediation, takes advantage of native plant-microbe interactions. So all plants have root systems that have niches for bacteria and other microbes to live there, and usually a symbiotic relationship, provide energy, protect against pathogens. The interesting thing about these plant-microbe interactions is that the plant will secrete these high-energy compounds that provide an energy source for the bacteria then, that allows them to metabolize these non-energy releasing metabolites. They can provide cofactors, surfactants. They can relieve environmental stresses by providing this very uniform niche for the bacteria. They can also serve as a biofilm anchor surface, particularly in water. So if you have like this duckweed or other pond type things, you're basically providing an anchor for bacteria to colonize. And they oxygenate the water as well, so you're not working with an anaerobic environment. So just keep in mind that as much as we talk about modifying the plants themselves, you're also going to be changing the interaction with the microbial community, and that itself can be engineered. So how can synthetic biology help? One way is to transform microbial catabolic pathways into plants, or, mammalian catabolic pathways. We know a lot about cytochrome P450, so we can take those from other organisms. You can enhance the expression of native pathways or enhance the secretion of degrading enzymes, things that are normally in the cell you can secrete into the root system. We can also create synthetic mutualist plant microbe interactions. I haven't seen any papers on that work yet, but stay tuned. So for example, overexpressing native genes. So these three enzymes, adenosine triphosphate sulfurylase, and I'm not going to try and pronounce the rest of them, basically they detoxify the selenium that a plant takes up. And you can see here that in this one study, when it's overexpressed in Indian mustard, you get a 4-fold increase in the ability to take up selenium from mine waste soils. So that's just overexpression. We wouldn't really think of it as transgenic, but it counts as transgenic to the EPA. So, taking bacterial genes and expressing them in, I think this one was tobacco, allows you to degrade TNT. So if you have these military contaminated sites, you've got TNT that you need to break down. What they did is they took a nitroreductase to take this NO group here and break it down into an NH2 group, which is much less reactive, much less toxic. And so again, these are your plant seedlings. You germinate them in flasks and just leave them in light for a while, I guess. It still kind of blows my mind. But anyway, so the wild type here looks pretty dead. It's killed, this is the highest soluble concentration of TNT. So it doesn't break down any of the TNT. And the product here, the ADNT over here, is what they're expecting to find from this procedure. Doesn't increase. This one clearly has survived after 14 days in the highest soluble concentration of TNT. It's done a very good job of breaking down TNT. You don't really see any of the product accumulating, and they weren't really sure why. They're thinking that this is, so there's no known way to take this and break it down, but you do instead conjugate it to sugars or things like that for storage in the vacuole or the cell wall, and they don't know what procedure there is being used. So it's difficult to detect what specific metabolite this is going into. Again, need for further research. We know that the TNT isn't there. We don't know what is there. Awesome, and we'll break for pizza. Okay.
I guess it's encouraging.
Or rather, now that you can make anything for free, what do you think you will make?
I still have to submit my ideas. I had some back in that meeting.
Okay, so in the interest of time, I'm going to keep going.
I'll go back to you.
Shut up!
Another method is to secrete transgenic proteins into the soil. And this is particularly helpful for compounds that aren't very soluble, aren't very bioavailable, and plants
It's
can't
delayed,
take
it's
up
delayed.
directly. So for example, this beautiful mushroom here is C. versicolor. It has a lactase enzyme that can break down BPA. Unfortunately, you can't get BPA into this mushroom. So they took this lactase enzyme and expressed it transgenically in tobacco with an export signal so that it's secreted from the root. And just as a reminder, because you're randomly integrating these genes to anywhere in the plant genome, instead of just one targeted insertion, you have to do a whole bunch of these experiments to see which genomic context is best. And so there are a whole bunch of replicas here, that's why. But you can see that compared to the control, you get much higher amounts of removal of BPA from the soil. And this is not even by taking it up. This is just secreting the enzyme and breaking it down in situ. And then not just storage detoxification, instead of having these radioactive sunflowers that you then have to figure out how to dispose of them, it would be really great if you could take these toxic compounds and break them down or make them detoxify them. So poplar trees, which are kind of an old world tree that looks like an aspen, they have these very deep root systems which allows them to get really deep into the soil and take up a lot of water. They transformed these trees with rabbit cytochrome P450 in order to break down trichloroethane. This also works on, I think, benzene, toluene, some really nasty stuff. But it shows 100 times higher metabolism of these nasty compounds. So this was actually a field trial. They developed these in the lab and then did a field trial for these. And they were able to show that this trichloroethane is broken down into the glucoside and stored in the leaves. But this doesn't account for the vast majority of the trichloroethane, and they weren't quite sure what was going on there. So they did a mass balance, which was a mass balance of the taurine, which was rather difficult to do in a field trial, I would expect. But they found that the wild-type poplars would take it up. They'd store it in the trunk as trichloroethane and things. But then they would also volatilize a lot of it. It would be released through the leaves. Whereas the genetic modification to include this cytochrome P450 allowed this to be broken down into less toxic variants, which meant that it wasn't excreted through the leaves and volatilized. So the chlorine balance found that there were primarily chloride ions that were stored in the tissues and in the soil, which means that the rest of the trichloroethane got broken down into organic compounds and either metabolized or released in a non-harmful way. Right, so moving on to stress tolerance, the largest thing we're concerned about right now is climate change in terms of increased heat, but we also have to think about what would the world be like if we could grow rice in salt water? What would it be like if we could not worry about frost resistance or frost destroying crops? And so those things are kind of under this umbrella as well. So you can think of metabolic stress relief. A lot of the individual single-cell solutions look at metabolism. And one way to do this is by increasing the concentration of metabolites within the cell. So that's kind of an osmoprotective effect. When there's heat stress, it helps keep the proteins properly folded. When there's cold stress, it keeps ice crystals from forming and permeating the membrane. So they took Arabidopsis, which is kind of the model plant. They expressed choline oxidase from Arthropacter globiformans, which accumulated this metabolite glycine betaine in the seeds specifically. So they specifically exported these to the seeds. And that allowed the seeds to sprout and grow at high temperature. So you can see here at 32 degrees, none of the wild type seeds grew. You still got some good germination in the modified ones. The same modification has been studied in several other trials for cold and salt stresses as well, and seems to be kind of a broad protective mechanism just by changing the osmolarity of the cytoplasm. In terms of organ level, most of us work in single-cell organisms, so we don't really think about organs, but plants, of course, have organs. So they found this ubiquitin ligase. They weren't sure what it was. They just kind of looked for something that helped with thermotolerance, and they found this gene HTAS in rice. HTAS is heat tolerance something.
Associated site, maybe?
Probably. And so they actually found out that it was a ubiquitin ligase, which is like, who would have ever thought that a ubiquitin ligase helps with heat stress? But they found out that the organ that was treated here is the stomata. So within their leaves, plants have stomata, which are like little pores that open and close to take in carbon dioxide, release oxygen. Unfortunately, when you open your leaf like that, you also let out a lot of water. And so you can have transpiration stress. Basically, you are losing too much water. And a lot of that's elevated in high heat. And so they looked through, I believe this is electron microscopy at the stomata, and looked at the percentage of stomata that were open or closed. And they showed that this particular locus, this ubiquitin ligase, somehow increases the amount of stomata that are closed or partially closed, and therefore decreases the transpiration stress. And you can see that before heat stress, they all look pretty much the same. After heat stress, you can definitely tell a difference between the wild type and the two genetically modified overexpressing plants. And finally, protein engineering. So I was told I couldn't include any of my own work, but this is what I'll be doing for my postdoc. So if you've got questions, ask me later. It's not my own work yet. But the most thermosensitive enzyme in photosynthesis is rubisco activase. And the idea is that if you can increase the thermotolerance of this particular enzyme, photosynthesis can continue at high temperature with high efficiency. So they took the rubisco activase protein from agave, which is a desert plant used to high temperatures, and expressed it in rice. And saw that it was, so this blue line here compared to the green line, it can last at much higher temperatures. What they then did is some domain transplantation experiments where they took the agave C-terminal domain and stuck it on a rice N-terminal domain and got almost as good heat tolerance. And then they did point mutations and found the one point that converted thermotolerance. So you can even do some protein engineering. Again, this is very, very difficult to do with single genome, or with the genetic engineering technology we have, where you just insert it randomly in the genome. And I'm running out of time, so I'm going to talk really quickly. This is the CRISPR thing I promised you. So cucumbers are really good for CRISPR because they only have 2 chromosomes. They're diploid. And so you don't have to backcross them forever and ever to get a trait to go through. There are lots of viruses that affect cucumbers and melons. One of them, so this family of Potiviridae viruses uses the elongation initiation factor 4. Now, in cucumbers, they have multiples of these that are somewhat redundant, but the virus only uses this particular one. And so they use CRISPR to knock out this gene. So this is a heterozygote, it's a recessive trait. This is what the virus does to the leaves. And you can see that just by knocking it out with CRISPR, you can get almost complete resistance, even though there are titers of virus in some of these. Right. There's also an effort to make perennial crops. So typically with wheat, corn, and most of them, the grains, you have to replant them every year. And someone said, wouldn't it be great if we could just not replant them every year? Have them develop this really big root system that reaches deep, gets more water, more nutrients, is more stress resistant because it has this deep root system, and not have to replant it every year. So there's conventional breeding of wheat and sorghum, back crossing it to wild varieties. And of course, because wheat is hexaploid and you don't know which traits are for yield and which traits are for perennial lifestyle, and they're all very complex, it's taking forever. They're estimating that sometime in the next 10 to 30 years, we might have a commercializable variety that might be 50% as efficient as modern ones. So the genes controlling this are unknown, but the genes affecting yield are largely known. And so if we were to use biotechnology, we could potentially improve the yield of wild variants. There's also the case for domesticating perennials. So taking natively occurring grains that are perennial and domesticating them for human consumption. Currently, there are 2 underway, Kernza and Sulfum. Sulfum being for sunflower oil, and Kernza as a wheat substitute. That's Kernza there with the very long roots. And it's actually been commercialized as this long root ale. So if you're interested in supporting the this kind of work, find it at your local Whole Foods. And biofortification, really quick. Biofortification allows you to improve the quality of foods. So there's this micronutrient deficiency where even if you have enough calories, you're not getting your vitamin A, iron, or zinc, and that causes really nasty things. 1 in 3 people are affected worldwide, and 7 million children die annually from this. So the idea is, why don't we take existing crops and increase the titers of iron, zinc, or vitamin A in them? Develop and distribute these improved variants, and it's a one-time investment. You distribute the seeds, you ask the farmers, next year pass on the same number of seeds to your neighbors, and instead of having to create this this fortification pipeline like we use for milk, you add vitamin D. Conventional breeding is the main thing here. Iron, beans, millet, cassava, sweet potato, etc. have been made. And it does work, so the maize...
That's important.
Yeah, the corn that expresses vitamin A actually works just as well as supplements to increase the vitamin A load in children. So this is currently going on. There are more than 150 biofortified varieties in 30 countries. They're now consumed by 20 million people annually. This is all through conventional breeding, and the main challenge here is to increase the throughput. So if you're using conventional breeding, you need germplasm banks that have a variety of biodiversity back cross in. You need to get better measurement methods to measure these in high throughput and use molecular markers. Synthetic biology, of course, we all know can help through things like protein engineering, importing in non-native pathways. None of this is new to you guys. Golden rice is a key example here. By adding genes from, I believe, daffodil, they were able to increase vitamin A expression and make sure that it got to the seed. But there's a problem. There's always a problem. Almost done. So there's this large resistance movement that affects all of what I've talked about today. Not just the genetically modified food, but also we don't want to have genetically modified sunflowers growing near Fukushima kind of thing. So what do we do now? Your job as people who know about this is to go educate people, to reduce the stigma of GMOs, demand, so consume products that are made with genetic engineering. That's one of the good things, I guess, that has come out of the genetic engineering labeling, is that you now know that it's good to eat M&Ms and support this. You can also boycott companies with anti-GMO policies like Chipotle. You can also advocate to U.S. Often sets the regulatory tone for the rest of the world and donate. If you have any questions, talk to me later.
Woo!
Thank you so much, Sarah. If you have